Hi everyone, and welcome to our screencast all about protein synthesis, how we make proteins. We share a lot of proteins with all the creatures that live on Earth. Worms, flies, plants have the same proteins that we do. Proteins in our fingernails grow 3 or 4 times as fast as the proteins in our toenails do. It takes more muscles to frown than it takes to smile, 43 versus 17. And of course, skeletal muscles are the most numerous muscles. We use 200 of them just to go for a walk. So what do you need to know by the end of this screencast? Well, you need to know the basic steps of protein synthesis. You need to know and understand the sequence of amino acids and be able to look at a chart to figure out what protein DNA is going to make. You need to be able to describe 2 environmental mutagens that can cause mutation and then explain how mutations affect DNA and can lead to genetic disorders. And we talked about a lot of that in our last screencast. So you're going to need to know some vocabulary words all about protein synthesis. So some old words, some new words, make sure that you know them all. We talked when we were discussing biological molecules all about proteins. Proteins have 3 functions. We'll talk about 2 here: structural and functional. So proteins like antibodies, proteins that are in our cell membranes, so stuck in between those phospholipids of our cell membranes. Some of them are transporters, some of them are receptors, some of them are enzymes. So lots of different proteins function there. Of course, hemoglobin is a protein, and enzymes are also proteins. We're going to be discussing enzymes with a whole unit of their own. So protein structure is determined by your DNA, and one little piece of DNA that codes for one protein, of course, is called a gene. And a gene is a piece of DNA, and that DNA determines what the primary sequence of an amino acid is going to be. So if you have a piece of DNA, the gene in that DNA chromosome will be copied as mRNA, and the mRNA will then be translated into the primary sequence of a protein. And we know from our protein unit that that primary sequence will then fold up into its secondary structure and then ultimately into its tertiary structure, its very specific 3-dimensional shape. So the gene in DNA ultimately determines what the shape of the protein will be, and because the shape determines the function of the protein, genes determine the job or the function of all proteins that function in our bodies. So one gene codes for one type of protein, and that one protein has one job or one function in the cell. So today we're going to be talking all about protein synthesis, and protein synthesis has 2 main steps. The first step is called transcription. And transcription is just making a copy of one gene in your DNA into a messenger RNA. The second step in protein synthesis is called translation. And translation is taking that mRNA, that messenger RNA, and translating it into a protein. So one gene ultimately makes one protein. So how does protein synthesis work? Well, I like to think of it as a play. So the director of the play is the DNA, the gene. And there's 3 assistant directors: the mRNA, the tRNA, and the rRNA. The mRNA stands for messenger RNA. It's the one that carries the copy into the ribosome. tRNA stands for transfer RNA, and rRNA stands for ribosomal RNA. It helps to make up the ribosome. We'll talk about all of those 3 as we go along today. Back to the play, the cast, of course, would be the amino acids forming a protein. The stage, where does it all happen? At the ribosome. So the ribosome is the stage, and the crew, of course, nothing happens in our body without the help of enzymes. So let's talk right now about the first act in the play, and that would be transcription. Trans means across, cription means to write. How do you remember that transcription is the first stage? I think about the monks. And monks back in the day used to take old copies of text, like Chinese text, and they would copy them word for word in whatever language, and they didn't know what it was translating into. They were just copying it to make sure that there would be copies for future generations. So transcription is just making a copy of one gene, and we know that that gene has the specific instructions to make a protein. So the gene is copied into messenger RNA. This happens inside the nucleus. Now DNA on DNA will code for a U on mRNA. T codes for A, C codes for G, G codes for C. If you take a look at this piece of DNA, ACG, G, C, T, 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 A, C, G, A, and you transcribed it into an mRNA, then you would get A codes with U, so U, G, C, C, G, T codes with A, CGA, A, U, and G, C, U again. So instead of T's, you do U's with the mRNA. So here we have the mRNA, and it has been copied from the DNA. And once the mRNA has been copied, it leaves the nucleus through the nuclear pores. So the process of making mRNA is called transcription. And step one is helicase will unwind the DNA, but not the whole length of the DNA, just one gene. And then you get complementary base pairing, but of course instead of DNA base pairs you'll get A, C, G, and this time U because you're making an RNA. Then you'll have RNA polymerase, and that will form the sugar phosphate backbone and check for mistakes. Now these steps should sound very familiar. They're exactly like DNA replication, except this time we're making RNA. The fourth step though is different. The RNA will detach and then the DNA will wind back up. So let's watch the process. Here you've got complementary base pairing happening, and the RNA polymerase is helping to form that sugar phosphate backbone. As you can see, the RNA is being built, and it's only going from the promoter to the terminator. It's only going from the beginning to the end of one gene on this blue chromosome we've got right here. So once we hit the end of the gene, then the RNA will be released from the DNA winds back up, and then the RNA goes off and it goes through the nuclear pore to the cytoplasm. So the steps, if you've got a piece of DNA here, step one, the DNA unzips. It does that with helicase. Step two, your complementary base pairs come in. So if this were A, then a U will come and join it. If this were T, then an A would come and join. If this were C, then a G would come and join, and if that were G, a C will come and join. So that's complementary base pairing. The third step that happens is your RNA backbone will form. So you got to join all those sugars and phosphates together, and RNA polymerase does that while it checks for mistakes. And of course, lastly, the RNA will be released and the DNA will wind back up. Let's take a closer look at that process. So how does the RNA strand form? Once the DNA is unwound, the A's will bind to the T's, the C's will bind to the G's. Of course, that takes energy, and then your sugar phosphates join together, and RNA polymerase helps with that. Because we're making an RNA, a U will join to the A's, and there's your sugar phosphate backbone again. And this process will continue for the entire length of the gene. Once the RNA has been completely formed, then those hydrogen bonds will be cut so the RNA can leave and exit the nucleus through the nuclear pore. So make sure you join me next class with all of your hot questions about protein synthesis and transcription.